Hello and welcome to all to our ISNTD Connect. We're here today, one day before World Malaria Day for our Malaria special, and we're very delighted to be joined today by Dr. James Duffy. James, you are Director of Drug Discovery at Medicines for Malaria Venture. Sorry.
That's okay.
And we're really thrilled to have you here today. I think obviously we've spoken a lot about neglected tropical diseases as per the WHO list in the last few weeks. And of course, touching a lot on COVID-19 as well. And so I think it was great timing and a great pleasure to have you today, speaking to us a little bit more about where we're at in terms of malaria and also what the future holds in terms of collaboration and drug discovery for open innovation in antimalarials. So we're really pleased to have the opportunity to celebrate in our own way World Malaria Day today with James. What's been interesting is that this day normally kind of comes with a lot of a certain level of cheer, and it's kind of a jovial moment to take stock about all the great advances that have been done in malaria. But unfortunately, this year, World Malaria Day kind of is preceded by a few stark warnings, whether it be from WHO or the science community, about what may lie ahead in the aftermath of the COVID-19 pandemic. And these are things we need to bear in mind. So in this slightly unusual year, I'll hand over to you, James, and hear a little bit more about what Medicines for Malaria Venture has in play to tackle malaria treatment, but also how you've been working in the field and with some of your partners.
Okufuma obushiku bw'amairo omwana alisa alwala omubiri we alisa akabasaana alisa afirwa n'okulya n'ebyakulya efa n'akiisa afika nakuli bakome kishinka bamupima n'omulopa bamusanga n'obulwere buntu bakimusanga nabo.
Severe malaria, if left untreated or unchecked, can result in death, especially within the first 24 hours. The majority of lives lost to malaria are those of children who live quite far from health services.
How do you save children who are living in remote communities who are suffering with severe malaria?
Here in Zambia, we're using an already existing form of antimalarial treatment, artesunate. In this case, it's in the form of a rectal suppository. This is a life-saving form of drug. The initial dose allows time for the sick child be transported from the remote area that they're coming from to the health facility where they can access more treatment.
Zambia is a very big country with about 752,000 square kilometers. Some villages can be as far as 50 kilometers to a health facility. If you have to get the child to the health facility in time, there must be a transport system in place. So these bicycle ambulances are a much better mode of transport because they need to be transported in a very, very proper way so that you don't complicate the condition.
By the time they are reaching the health facility, at least they are stabilized. Now at the facility, we usually give injectable artesunate in order to complete the treatment.
Lero nakireeta omwana ku kipataala nakimuleeta n'okumundapa bamundapa n'omwenso nshikwetewa kwe batikutyawo naika.
Since the introduction of this antimalarial drug, we've seen a huge reduction of death in children under 5 in remote areas.
By improving access at community level, we can save more lives, which has been proven already in Zambia. Now that there's a World Health Organization quality-assured rectal artesunate suppository, this has the effect of improving our fight against malaria. Currently, 29 countries have introduced this form of treatment. This is one of many innovations that we need in the next 5 years in order to beat malaria.
So thanks a lot to Marianne and the team at ISNTD for giving me the opportunity to speak today. Now, obviously, those of us, especially those of us who live in the Northern Hemisphere, are well aware of the current pandemic situation, but we know that almost half the world's population for all their lives has been living under the terrible burden of a pandemic that disproportionately affects the most vulnerable of populations. In the video, I wanted to show a little bit of the human reason why we actually do the work and why we're so passionate about it, and some of the contributions we've made. Throughout the presentation, I'm going to delve a little bit more into the science. I actually work in the discovery team within MMV, so my actual focus is the discovery and development of new medicines, as opposed to actually the clinical trials and actually ultimately getting them to the patient. But so we're going to focus on that end of the drug discovery process, but we'll touch on the whole length of it. So please do ask any questions, and I'll try my best to answer them. So let's try and start the presentation. As Marianne said, tomorrow is World Malaria Day, and we really like to use this day as an opportunity to not only celebrate our achievements, but also to motivate ourselves for the fight that remains ahead, and to highlight to our funders, to the general people, to the politicians, that this is a fight that is not over. This is a fight that's not won, and this is a fight that needs to continue to be front and center of people's minds and attentions, in terms of funding and in terms of research. So, as I said, I really don't need to dwell on the statistics on the slide here. I think you're all very familiar with the burden that malaria causes to almost half the world's population. And as I said in my introduction, it disproportionately affects the most vulnerable populations, pregnant women and children especially. And as well as the human impact, the economic impact of the disease is immense, and really is a reason for wanting to address this terrible burden. And this slide now is probably, as Marianne perhaps alluded to, has a potential for being horribly out of date. What we're looking at there is where we are standing. This slide was put together in 2019, so essentially we're at the line on 2020, where we are with around about 400,000 deaths occurring annually at the moment. And the authors of this study predicted, modeled what would happen if we continued on our current trend of reduction, and you can see that, where we'll be way above the what we could achieve, and what the WHO and organizations and the countries endemic to the disease want to achieve, which is elimination by 2040. You can see the worst case scenario, which is simply what would happen if we do not develop new antimalarial drugs and resistance does emerge in the field. And it is inevitable that resistance it's a biological inevitability that resistance to the medications will at some point that are used on the front line will at some point occur. And this is what will happen if we see resistance to the frontline medications. And then the red line is really the inspiration. It's what we could achieve if we are able to strengthen the existing malaria control tools, management, use of the available drugs, to develop new medicines, to develop new vaccines, to improve our mosquito control tools, whether it's bed net sprays, whether it's insecticidal sprays, whether it's insecticidal medicines. And the only way that this can be achieved is with financing from both malaria endemic countries, non-malaria endemic countries, and donors. And in order to follow the path of that red line, we're looking at needing in excess of $2 billion annually additional funding, but the rewards for achieving that on both a humanitarian and an economic scale certainly justify that level of expenditure. However, we are very mindful that due to the impact of the current SARS-CoV-2 pandemic, we may well see a spike, or we're predicted to see a spike in 2020, 2021, back up to potentially doubling almost the case numbers, due to an impact not just on the distribution of bed nets, the seasonal malaria, chemo prevention medicines, but also in terms of actually supply of rapid diagnostic tests, which may put increased demand on a supply chain that's already under stress, whether it's stress due to the closure of certain sites producing it, whether it's stress to repurposing of antimalarials, or whether it's stress that clinicians will have to actually start using antimalarial medicines on an assumptive basis rather than actually following a diagnostic test. So all these pressures we're well aware of, and could lead to a worsening of the situation in malaria if the world is not incredibly mindful of the potential challenges. So a little bit of background about MMV for those of you that aren't aware of us. We were founded in about 20 years ago. Last year was our 20th anniversary, and our mission was to discover, develop and deliver new effective and affordable antimalarial drugs. And this is solely focused at vulnerable populations in disease endemic countries in terms of our target population, pregnant women, young children, in terms of our cost structure, making the doses and the treatments affordable. And as with so many other infectious and neglected and tropical diseases, the drug market is enormous in terms of the number of patients who are crying out for these medicines. But it's, in terms of pure profit, it's small, which makes it very difficult for pharmaceutical companies operating under the traditional drug discovery and development model to be able to justify this to their shareholders. So MMV raises funding from governments, from philanthropic organisations such as the Gates Foundation, and we share the costs and the risks of doing this, which makes it possible for us to carry out this research with both academic groups and with pharma organisations. In the last 20 years, we have had some notable successes in co-developing medicines that have saved lives. And here I've shown 4 of our 11 medicines that we've actually had approved for use in patients. The first, coartem dispersible, is treatment medication. So this is the frontline medicine for non-severe malaria. And is used in a pediatric formulation. So essentially, coartem, for those of us who've taken it, is an incredibly bitter tasting medicine, and it's unpalatable to young children, certainly in a pediatric setting. So with help from Novartis, we developed a sweet fruit flavored formulation of the medicine. And over 350 million courses of this have been delivered, saving almost 1 million children's lives, it's estimated. The second 2 medicines, artesunate injectable and artesunate rectal, were highlighted in the video that we saw before my presentation. So these are for the treatment of severe malaria. Injected is normally given within a clinical setting and used to rapidly bring down the parasitemia. But as you can imagine, with an injected therapy, it is used in a clinical setting generally, whereas the rectal artesunate is able to be used by health workers and in the community setting, and stabilizes the patient, allowing them to buy some time to be brought to hospital where they can receive standard of care therapy. And SPAQ, this is really an example of where we're using life cycle management. So SPAQ is a sort of, if you like, a medicine that is currently not being used so much for treatment, but we use it for seasonal malaria chemoprevention. So in this case, doses of the medicine are delivered to young children, and they take it prophylactically. And in this case, we've been able to reduce uncomplicated and severe malaria episodes by 75%. Which has led to a great improvement in life-saving medicine. Okay, so that's really the picture of where we are. We've made great progress. We think that with increased funding, that we can continue to drive down the curve and work our way towards malaria elimination by 2040. I've said that we've got some great medicines that we've developed that are effective, but there are challenges, and challenges do remain. when we are developing new antimalarial drugs, and we do need to develop new antimalarial drugs. And here I sort of show them from a drug discovery perspective the three things that we really keep at the front of our mind when we're thinking about new antimalarial drugs and what we need to do. So the first point is the drug combination. So we develop combination therapies to protect against resistance, and so the combination therapy needs to deliver a cure. So this is killing up to 10 to the 12 patients in a parasite. No, 10 to the 12 parasites in a patient. So this is to sort of orientate your mind. The ACT, so the frontline therapy, clear about 4 log units in 48 hours. So that's possibly, we have to go for 3 times 48 hours coverage of the drug to, in the worst case scenario, to actually clear the patients. We have to achieve that killing in every asexual life cycle until all the parasites have gone. And that, we need 3 to 4 life cycles of exposure, so 6 to 8 days from an oral dose. The current treatments are taken 3 times a day. And ideally, we would love to develop a medicine that can be taken once. This would allow the health worker to administer the medicine and then leave to go to the different patients, to a different village, to a different setting, and not have to return to give a second dose, or alternatively leave 3 doses and the medicines do work. So those of you who've taken them know they work, and after the first or second dose of the medicine, you often feel much better. And there is the opportunity for the treatment courses to not be completed, and for the possibility for drug resistance. Talking about resistance, the medicines must be efficacious against all known field resistance. Malaria medicines, the ACTs, for example, work well in most parts of the world, certainly within Africa. However, in Southeast Asia, they are failing, and they are the number of treatment doses that are having to be given is being extended. And we are very concerned that this resistance will eventually reach Africa. And we will need new medicines in the medicine cabinet that we have ready to go when this happens. And if that wasn't enough of a challenge, these have to be developable at a cost-effective $1 a dose, $0.50 a dose for children. And they have to be fixed dose. We can't, we need to have them ready to go. So the dose that we give covers all scenarios, and they have to be child-friendly. Ideally, they have to be maternal-friendly as well. So we have been working, and this is, I have another slide similar to this one, which shows what our pipeline was like in 1999 when we started, and we didn't have anything in the pipeline. And that is globally, there was no new medicine in the pipeline for malaria at that point. Here we have 14 drugs that were all in clinical development that have been discovered and developed in partnership with MMV, and our pharma and our academic partners have all developed these compounds and identified. And as you can see, we have compounds in clinical trials, we have compounds in preclinical trials. We have several new mechanisms of action that have been discovered. All compounds are effective against the asexual blood stage of the malaria parasite. That's that sort of red blood cell looking icon. Some, where you see that shield, are have potential against the liver stage schizonts of the parasite, and some have transmission blocking potential, i.e. they're active against the gametocyte stage of the parasite. However, we have no new medicines in our pipeline that are active against the relapsing form of malaria. The good news is we were able to, in partnership with GSK, launch a new anti-relapse medicine, tafenoquine, last year. However, this is building on the primaquine, which was the standard of care prior to tafenoquine, and again has a liability for, it causes hemolysis in patients that are deficient in G6PD enzyme. So you need to have a diagnostic test as well. We would like new anti-Vivax medicines. And there are reasons that I'll talk about why we haven't actually been able to develop these. Okay, and how are we able to do this, and what are we thinking about next? And so what I'm going to talk about in the next couple of slides are some of the new assays and how we identify new targets. And the quote for here is "Ask the parasite." So the Maldor consortium or Malaria Drug Accelerator Consortium, is a target-guided drug discovery platform. The majority of those compounds you saw on the previous slide were developed through phenotypic screening. This is taking parasite-infected red blood cells and testing medicines against them. And you look for parasite growth inhibition. So it's a simple but functionally effective assay, and it translates well to in vivo models, and it translates well once you bring it into human. But you don't have so much information about what target you're actually affecting and what your potential off-target safety issues may be. By doing target-based discovery, we have the potential for doing cell-free assays. We have the potential for evaluating and mitigating against resistance. We can do high-throughput screening. We can do structure-based drug discovery, and that's what this consortium is really doing. And we feed in all sorts of compound libraries and the hits from compound libraries. The consortium, which is a group of Bill & Melinda Gates Foundation funded organizations, including academic groups, pharma groups, some based in North America, some based in Europe, some based in Africa. This consortium are really the machine for finding these new exciting malaria drug targets, and they all have their own expertise, whether it's in whole genome analysis or assay development or doing reverse genetics, and they really have been founded to be a sort of an engine room for new and exciting drug targets for researchers. So when I said we haven't got any new drugs targeting Plasmodium vivax, why was this? Well, actually, the reason is, in terms of the normal sort of drug discovery paradigm, and I come from a pharma background, so we were unable to do screening against Plasmodium vivax. We were unable to culture Plasmodium vivax in vitro. So really, the only way that you could look at the assay was either in a surrogate system, such as say a murine model, which isn't the same as the human form of the disease, or you were having to hope that to test really against in an ex vivo setting against P vivax affected blood. And that wasn't really a liver stage model. And what we wanted to do was to have compounds that were active against the hypnozoite, so the relapsed, the dormant form of the parasite. However, a group at the University of Georgia, led by Professor Dennis Kyle, has now developed a 384 well format liver stage assay. So this is using primary human hepatocytes that they are stable for out to 3 weeks. So they can look at the effect of your compound on hypnozoites after 21 days of culture. So we've screened 330,000 compounds against this assay, and we have found new hit compounds. Now, 30,000 compounds, for those of you from a pharma background who are used to screening millions of compounds, isn't a lot, but is the first steps in actually having a drug discovery enabling P vivax liver stage assay. So I thought I would just, for the medicinal chemists in the audience, I'd show a few structures of some of the new candidates and where they come from and how we're feeding compounds into the pipeline, and where these new drugs come from and why we're so excited about them. So the first one, this is an interesting story, and it's something to remember, I think, for all of us involved in drug discovery, that we have to keep an open mind, and sometimes we have to look backwards as well as looking forwards. So JPC, this was developed by Dr. David Jacobus and his team at Jacobus Pharmaceuticals, and David Jacobus was working at Walter Reed in the 1970s. And identified the compound you can see on the far left of the screen, so this Walter Reed compound. At the time, this was taken straight into volunteers, and at high dose, it was effective, but it had issues. It caused torsade de pointes, so it was a hERG inhibitor, and it wasn't particularly efficacious. So at the time, it was dropped. There were, malaria. There were medicines that were available, and chloroquine, the sort of mefloquine, and people weren't so interested in it, so it got parked. But David never lost sight of this, and was always trying to develop it and address the issues. And a few years ago, he approached MMV again and said, "Look, I've still been working on this compound. Are you interested in it?" And you can see the compound JPC3210 on the right hand side, where they've improved potency. They've got it's active against drug-resistant parasites, it's long duration, and it's got a reduced herd liability. So this was a compound that was approved by our scientific advisory committee last year, and is going to go into clinical development as soon as possible. This is active against the blood stage of malaria. The second, this is perhaps a more traditional, to say traditional, at least if you look back to the sort of the 1980s, 1990s, and the 21st century approach to drug discovery. So this is carried out by GSK, the MMV, and the Wellcome Trust who funded this. And GSK have screened their compound collection. So fortunately, many pharma companies are happy to screen their large proprietary compound collections against neglected disease targets such as malaria. And we have a high throughput assay for the blood stage of malaria. We can screen millions and millions of compounds without too much effort and at relatively low cost. And GSK did this screen, and they identified the compound you can see there below. And this is a new target, PFACS10. It's rapid killing. It's got a low predicted single dose, and it's safe. So this again will go into human studies. So finally, what I want to end with is some of the tools and this way we're thinking about how we can help the community continue to find new drugs, and how we can stimulate thinking, and how we can really move the approach onto the next chapter in the book. So the first slide here, there's a lot of information on this slide, but essentially this is a machine learning approach. So we've got a lot of data on malaria activity. We've screened probably somewhere around about 8 million compounds with a number of organizations, ourselves, some of the pharma companies, and we got them all together, and we essentially, in this approach, each of them develops a QSAR model, so a computational model, to predict the active compounds that they had compared to their inactive compounds they tested. All of these models kind of get fed into a central model that then it doesn't matter what approach they've all taken. It makes a consensus QSAR model, and this work has been completed by a group at the EBI in the UK, and this will be shortly released as a web-based tool. So any researcher can, based on the data that we have from all of these different partners, will be able to submit their compounds or their designs of compounds or their compound libraries into this online tool, and it will predict whether they're likely to be malaria actives or inactives. And we're doing a small piece of work around this, just about pharmacologically validate this. We did a piece of work a few years ago that did show this model works, but that version of the model was not open access. And we've now made an open access version of the model, and we're shortly going to publish these results. Another piece of work that we're really keen on at the moment is, wherever possible, trying to reduce the number of animal studies required. As I said, there's a great correlation between the in vitro data and the in vivo data in malaria. And normally how we would run the studies in the traditional paradigm, we test in vitro, we test in what we actually have as a humanized mouse model for malaria, and then eventually we'd actually test in volunteers. So this is our normal cycle. However, we've been doing some modeling and some additional work, and we believe that we can actually predict the likely efficacious concentration of our antimalarial drugs using the in vivo data. Using the in vitro data, sorry. And the graph you can see on the right is the correlation of some actual marketed antimalarial drugs based on using the in vitro data, and also based on the predicted in vitro data correlating with the actual experimental human data. And we have a reasonable correlation, and we think that we can really start to accelerate some of our projects and reduce the number of animal experiments that we need to use. And so I think my last two slides are really for any researchers in the audience to know that we have some tools, and this is not just tools for malaria. This is tools for anyone working in infectious or neglected and tropical diseases. So you may be aware that MMV, in this case in partnership with DND, our friends at DNDi, have with sort of slightly timely, made available a select of compounds. So the thinking behind this was lots of researchers have great biological targets, they have great assays, but they just don't have the tool compounds to probe these assays. And we thought we could plug the gap by making sets of compounds with known activity against one or more diseases or organisms available free of charge. And the latest, we have done this with malaria, we've done this with other pathogens, and we've now done it, in this case, the pandemic response box, which is antivirals, antifungals, and anti-infectives. And they are available for free to any researcher. The only obligation is that you agree to make the results publicly available. And the example below is just to show you that you can get something unexpected from screening these bioactive compounds. And here's the case. We had some of our groups screen them against the malaria. We weren't looking for malaria active compounds. You can see the compound on the right is actually, the far bottom right is actually tefenoquine, so perhaps we would have expected that to be active. But you can see we got antimalarial hits from screening a set of compounds that weren't associated with antimalarial activity. So this is something that we make available. And for those of you who have larger capacity assays, we also purchase compound libraries through the help of our generous funders. And recently we've purchased 2 high quality, diverse and novel compound libraries with good calculated physicochemical properties, no structural alerts. And we have copies of these that we again can make available to researchers for testing against their own particular disease of choice. Again, the only obligation that we have for this is again to make the results publicly available. So that is a brief sort of walk through what MMV do, why we do it, how we do it, and what we're going to do in the future, and why we still need to be doing this in the future. So all that remains is for me to sort of thank you for joining us on this call and for participating in World Malaria Day, and to our funders who've provided the financial support that have allowed us to do this research. So thank you very much, and I'll try and answer any questions.
Thank you very much to you, James. And I think everyone would join me in giving yourself and MMV in general a huge congratulations for these 20 years of incredible accomplishments in malaria drug discovery, but also control, partnership building, and then ultimately increasing access to those really in need on the ground in endemic countries. So a fascinating presentation. Thank you very much. And so for me, you've been in this field for over the 2 decades. And I think we might wonder, our first question to you might be, James, in your opinion, what would be MMV's greatest accomplishment out of all of these that you've listed and all the projects that are still going to develop and go ahead? What really stands out for you?
I mean, I suppose really you can't look beyond the numbers of lives saved. I think that in terms of any medicine, in terms of any medicines researcher, in terms of any clinician, the real benchmark that we hold ourselves against is numbers of lives saved. And I think, if you add together all the different medicines that we've worked on and the estimates of the numbers of lives saved, it's well over 2 million lives. What possibly even something, I think the last time I looked, it was about 3.2 million lives, and that is an incredible achievement that I'm a very, very small part of, and that I think is our greatest achievement. In terms of my own particular kind of, if I wanted slightly other slant on it, I think it's actually I'll give a story. When I started in the pharmaceutical industry, everything was very siloed. Everyone did their own research in their own organization. They did their own results, they did their own clinical trials. There were some areas we collaborated on, but very much the thinking was develop your own medicine. This has been broken open by a number of organizations, of which is only one. And the thinking now is that we can do more together It's the only way that we can do this research, the best way that we can do research. Malaria is a great example because there are so many different assays that you need to do, so many different pieces of the jigsaw puzzle, but no one organization can do that. And the only way you can do that is collaboratively. And you get where they're faster, you get there more effectively, you get there cheaper, and you deliver the medicines. And that's really, I think, you know, bringing together those partnerships is the thing that really has sort of inspired me. So 2 answers for the price of 1.
Yeah, they're both rolled into one, and I think that definitely makes sense. And I certainly want a legacy of MMV will definitely be to set that kind of tone for scientific collaboration. And it's become even more apparent in this time of the COVID-19 pandemic, but you know, the strength of the science and the research kind of work in the laboratories is actually totally connected to what happens out in the field. And so your approach really ensures that it remains very strong and connected all along the route, thinking about the supply chain as well. So very inspiring. And in fact, a lot of our attendees are giving you a very big thank you, James. Jeffrey Makenga, thank you for a great talk. Aline Nascimento is tuning in from Fiocruz in Brazil, saying thanks, James, for sharing the perspective for malaria disease treatments. We've got many more thank yous.
Thank you.
Ponzi, thank you for the nice presentation. Stephanie de Piquer as well. And so within all those, a few questions. So I will just take the first one is from Kamaluddin Benalal. What is the mechanism of some moleculars which act on blocking transmission of the parasite? That's quite a...
Well, I guess in terms of the actual sort of molecular targets, there's a number of molecular targets that we've identified that do have activity against the sexual stages of the parasite, and either kill the parasite or render it sterilized, so that when it's actually taken, the gametocyte's taken up into the mosquito, it can't actually undergo gametogenesis. So I mean, there are a number of mechanisms that we have, you know, PI4K, EF2. But in terms of a sort of broad phenotypic mechanism, generally the one that we will see will either be gametocyte killing or gametocyte sterilization, and normally it's gametocyte killing.
Brilliant. Thank you. Helena Lyotha, tuning in from Indonesia, is asking, do you think that vaccines development will be the effective way, since malaria has different stages and variations of infection?
Well, that's always a very relevant question. So thanks for that one. I will give one caveat before I answer my question. MMV, and I probably didn't say this at the beginning, we are only responsible for small molecule drug discovery. So you know, your ACTs, if you like, as opposed to vaccines. Other organizations are responsible for vaccine development, and so my opinion is a little bit as a outsider rather than someone who's actually working on them. In many ways, you know, vaccines will be the you know, we do feel it'll be a crucial piece in the jigsaw puzzle. What the split is between vaccines carrying some of the burden and small molecules carrying some of the burden, how much we can actually develop other biologics to actually, you know, complete the picture, remains to be seen. I mean, obviously, it goes without saying that the vaccine development within malaria has been incredibly difficult. You know, you only have to look at the efficacy of the current vaccines that are available to see there are many challenges in developing a vaccine that's effective against all stages of the parasite life cycle. In the same way, it's difficult to find small molecule therapeutics that are effective against all stages of the life cycle. I really think that if I can answer the question, the message will be we cannot just rely on one therapeutic intervention for this. They will all have to be done in partnership. And as I say, in terms of the long duration protection, it will be a combination of vaccines or of probably in long duration injectable antimalarials. So some combination of the two. It may even be biologics, but we have very little research into that at the moment.
Excellent. So on a slightly different note, Selvi Subramaniam was asking, first of all, thank you for the great presentation. Is it possible to prevent or delay the spread of anti-malaria drug resistance?
Well, that is an excellent question. We like to believe that it is possible. If you talk to the groups that really do study this in depth, they will say that the breakout of artemisinin resistance from Southeast Asia has been slower than it was with the breakout of chloroquine resistance or mefloquine resistance after that, due to monitoring, due to actually making sure that the patients are receiving their full treatment courses. How do we actually really slow this down? Well, there's two ways. Or maybe they're really the same way. One is that we use combination therapies. We will not see, I don't think that for treatment, no health organization from the WHO down to the regional health organizations is going to promote the use of monotherapies. So we will always have combination therapies where the hope is that the partner drug provides protection for the other drug in the combination. So the other way that we want to do is we want to look at single dose administration. So if that single dose provides a cure, there's no potential, there's less potential for people to take suboptimal doses. And the third is we do focus on compounds which have we find very different. We call them irresistible. Now, whether that's strictly speaking true or not is a sort of a biological question. But within the lab setting, we do in vitro resistance selection studies, and we focus on compound series where we're unable to generate resistance mutants in the laboratory. So we do have strategies for trying to minimize the risk of drug resistance. But drug resistance is a risk that does remain. But we try and monitor it in the lab and in the field.
We've got another question from Stéphanie de Pica, who I think is tuning in from Bolivia, La Paz. So again, this isn't aimed at the medicinal. Oh, can you hear me?
Yes, I can hear you.
Okay, yeah, okay, super. So this is aimed at more than the medicinal chemist in you. What do you think about mass drug treatment for asymptomatic people, and which drug would be better in that case?
Well, I mean, really, so I'm very happy to give my opinion on this, and I don't think it's particularly controversial, but I would like to say that I'm not particularly an expert on that end of the field. I think from my perspective, the real challenge of mass drug administration to asymptomatic people, beyond the sort of ethical considerations of giving drugs to people who potentially don't have, who don't have the disease, from a practical perspective, is really demonstrating the safety. I mean, the safety is absolutely paramount, and so to actually move a drug to that kind of confidence that you have to be able to give it to anyone, whether or not they have the disease, is a significant challenge to generate that amount of clinical data and have that confidence that you are going to be safely giving drugs to people who are young children, who are pregnant women, who have comorbidities. That really is going to be a challenge within that setting, especially when we're going to be at the end game of malaria, where the actual number of cases may be very low. So I agree. I think it's certainly far more complicated than just simply taking our best compound, making tons and tons and tons of it and giving it to everyone.
Absolutely. Catherine Brown is saying, thank you for this excellent presentation. Could you please expand a little more on the predictive capability of your vitro preclinical model, and whether the data you show is for a specific compound class and or is being applied with success to other pathogens?
Okay. So we have two models I talked. One was predicting in vitro activity, and that's really based on the asexual blood stage malaria. So the actual model is using phenotypic data, so it could be against a whole class of different molecular targets, and the actual structural types are wide and diverse. That the data set comes from millions of compounds. So that model and that approach, this sort of Bayesian learning, and I really, I'm not a computational chemist. I'm a sort of a lab-based sort of synthetic medicinal chemist. But the computational approach is robust for predicting, for enriching the prediction. So I think, when we first did the model, it was about a 10-fold enrichment over just randomly selecting compounds. So that works. Now we are talking, because this work was actually funded by the Gates Foundation, about whether if we had data sets from other pathogens, tuberculosis or leishmania, schistosomiasis, but we had big enough data sets, we could actually just simply use the same model. And the thinking is that we could. So I think the answer to your question is yes. I think it could be applied.
Great, thank you. A question from Freek de Haan. Great to learn about the progress in the antimalarial pipeline. What is your vision on combining currently used drugs into triple ACT as an emergency response to the emerging drug resistance, as long as true new drug compounds are not available?
Mm-hmm. Yeah, I mean it's a great point, and I think the limitations for that will really be around cost and if you like, the pill burden. If the compounds are efficacious and the dose is fairly low and the cost is fairly low, then it's really a technical formulation challenge, which I think we're capable of solving. If the pill burden gets high, if people are having to take grams and grams of medicine multiple times a day, then it just, rather than it being scientifically practical, it becomes sort of a clinical situation that isn't practical. When we were talking, and I was talking about the coartem dispersible, it's a great problem, it's a big problem that we have when we're sort of giving some of the investigational antimalarials to young children, and they have to, and you have a child who's 4 years old, and you're asking them to drink half a litre of syrup. It's very difficult for them to do that. If we were doing that with a triple combination, then it would be equally hard. If we can find the right partner drugs that allow us to make these low dose, palatable, low cost formulations, then yes, I think it's certainly something we'll investigate.
Great.
And obviously, sorry,
Jeffrey McKenga is asking why.
Please.
Oh, please.
I've finished.
Okay, Jeffrey. Oh, you finished. We're so polite to each other.
Yeah.
Jeffrey McKenga is asking, why do most malaria drugs work on the blood stage of Plasmodium falciparum? And do you have any candidates against cerebral malaria?
I guess treatment. Yes. So I mean, I guess treatment of non-severe, if you like, malaria has always been our focus. That's been the sort of the biggest patient population looking for the medicines from the beginning of MMV. In terms of actually developing drugs for severe malaria, we've always, rather than developing new medicines, we've always made use of our existing medicines in our pipeline. So, for example, artesunate, and we have other medicines in our pipeline that may actually be more suitable for use in severe malaria settings. They have to have the right physicochemical properties and the right, so you can actually formulate them the right way normally to give them intravenously, because if the patient is obviously in a coma, then it's harder to give the medicine orally. So they have to have the right properties to allow us to formulate them the right way. They obviously have to have the right rate of killing to allow us to bring the parasitemia down rapidly. But we've normally done that from our blood stage assays. I mean, they have to be active against the blood stage of malaria anyway. And why is our portfolio probably focused on blood stage malaria? Again, beyond it being the real sort of, at least when we started, the real treatment bottleneck has probably been, and probably because that was the treatment bottleneck, we spent more time developing the assays. And because we have more assays around that stage of the parasite life cycle, we can do more screening and we identify more hits. So it's a kind of chicken and egg answer that was led by that being really highlighted right at the beginning of MMV as being a critical need that we needed to plug, was the treatment of non-severe malaria.
Interesting, thank you. James, we have a couple more questions for you, if you have time to take those.
Of course.
Yeah, okay, thank you. So a question here from Zadil Nare. On the topic of resistance, is MMV testing combinations of the novel compounds to test for synergistic effects?
So yes, indeed, indeed we are, and we actually we actually have the way that we used to do our drug development was that we would develop and have approved 2 monotherapies, and then we would start the combination work. We're now actually starting to evaluate combinations in the clinical development phase, and even in our volunteer studies. We can even do it preclinically in our humanized mouse models to look for synergies, to look for slowing down of requiescence. So yes, we are.
And finally, James, you had a question. What were your thoughts on the following? A question from Winifrida Ponzi from Ifakara. I have noticed that in our country, most people, especially in rural settings, whenever they have a fever, they usually think it's malaria and just go to the pharmacy to buy antimalarial drugs without the consultation from a doctor. And do you think that this might be one of the causes of antimalarial resistance? And if so, what are your thoughts on what might be done?
Well, it's quite possible that is. I mean, you know, the worst thing would be if they were taking counterfeit medicines and they had malaria, and by getting by getting fake medicines, they weren't actually getting the treatment, or substandard medicines, that they weren't getting the dose they actually needed to treat the disease. If you know, you can still buy monotherapies in some over-the-counter settings, and they again add to drug resistance, but they tend to be the older antimalarials, so there's already resistance in the field there. How can we counter that? I guess education plays one part, but certainly access to medicines plays the biggest role. Access to affordable medicines is really the only way that we can allow people to affordable medicines and potentially diagnostics. So one is the healthcare infrastructure, one is having the medicines to treat the patients that come through that system. And it's been it's always one of the, you know, as you well know, I guess everyone on the call, is one of the, you know, the most distressing features about malaria is that it is by itself a treatable disease, and there are lots of reasons why we need to develop better medicines. There's lots of reasons why we need to develop new medicines, but essentially, you know, we do have the treatment tools available, and if we can get the right drugs to the right people at the right time, then you know, we've fought the hard part of the battle.
Absolutely. Donald Sanigo was asking about the Pandemic Response Box initiative. I think, Donald, you've written here that you missed part of the talk. Just to let you know, and also as a reminder to all our attendees, that the webinar is available on our YouTube channel. This will be posted probably in the next few hours. So if you feel that you've missed anything, please don't hesitate to go back to our replay on the channel. Also, I would invite you all to head to the MMV website, where there's a lot of information and detail about these boxes, the pathogen box, pandemic response box as well. James, perhaps you might want to give a few words for Donald about the pandemic response box, sort of
Well, yes,
what it entails, and also.
If any of you want copies of the pandemic response box, you can go onto the MMV website and request them. I mean, it's a very simple form, like you would fill in if you were, I don't know, ordering or doing online shopping or something, and we arrange for it to be sent out. It's, as I say, everything is free of charge. All you simply have to do is give your contact details and what bit of research you want to use it for. That's all you have to do that, to get that. If you want any, as Marianne said, if you want any more information, that that I can make these slides freely available, and you can contact me. I think my email address is probably on the MMV website. Otherwise, if you use any of the sort of normal sort of social media channels, you can probably contact me through Twitter or LinkedIn or anything like that. So please do contact me if you have any other questions or need any other information. We're always happy to help.
Fantastic, and I think that is definitely true. You're always happy to help. This has really been in the DNA of MMV, this extremely multidisciplinary, multi-partner collaborative model. So for that, we thank you very much as a community working in this field. Just perhaps to wrap up, James, I might ask, what are your plans for the future? Are there certain types of partnerships that you're looking for, or and where would you like to see MMV and your work in the short term, medium term? I'm assuming COVID's kind of put a hold on a lot of things, but beyond that, what might be your plans for the future?
Well, I guess the COVID SARS has diverted our attention, and obviously we've got some involvement in some of the clinical work that's going on around that, and giving our expertise where appropriate. I think our main focus at the moment is trying to manage the potential fallout from that in terms of malaria. That remains our real focus. Once life returns to normal, I think in the short term, we just continue to fill the pipeline with new medicines that really do address the unmet needs. We've got a really big focus on at the moment about mothers and babies, and that being 80% of our target population. It is a sad fact that really still our drugs are very much designed for people like me, so a sort of a European male, with no other comorbidities, when actually, and that's the pharmacokinetics, that's the efficacy, that's rather than actually being pregnant women or women of childbearing age and young children, especially malnourished children, especially people with comorbidities, really having drugs that really are suitable and focused on that patient population is where we're going in the next few years. And that will be really the key thing for us is, we're not developing drugs for Western tourists. We're not developing drugs for the military. We're trying to develop drugs for the people who actually take the brunt of this disease. And that's really got to be our focus. Longer term, I hope to be out of a job.
Fantastic. Yeah, that's what we all hope for, right?
Yeah.
That's a very positive message to end on, James. So we will definitely be watching this space and wishing MMV all the best for the next 20 years. And in the meantime, you've had lots of thank yous from all our attendees, particularly Raveen Pandey saying, thank you very much, James. I'm not from a pharmaceutical background, but your presentation has been very informative. So
thank you all very much. And thanks for all your comments and feedback. It's been an absolute pleasure to virtually meet you all.
We'd like to give you a last very warm thank you, James, for joining us today, and wishing you all the best with your work at MMV, and we'll definitely keep in touch.
Okay, well, thank you very much. Okay, have a nice rest of the day, everyone. Bye.
Yes, stay safe, everybody. Take care, and hopefully see you next week. Thanks again for everyone's time.
Okay,
bye. Bye.